Mr. Nelson? Mr. Nelson is joining us. He was giving us a few minutes for us to join, now that we took a bit of time to join. So give him a minute.
Good evening, everyone. I'm happy to see that you're here. So we had decided to give at least up to around 8:15 so that everyone gets a chance to join, and if not, we'll just proceed. So we'll just begin today's lesson. When we had a lesson, we talked about a couple of things. Some of the things that we talked about were some things to do with cell specialization, and we went ahead and said that cell specialization is the modification of a cell so that it can be able to perform some specific functions. And we said that specializations are basically adaptations or modifications. Okay, so we said we have different types of specialized cells in both plants and animals. And for example, as you're seeing on your screen, we give examples of specialized cells in plants. And we said we have, like, for example, root hair cell, which we said its importance is to increase surface area for absorption of water and mineral salts. So therefore, a root hair cell is specialized by having this extension that you can see there, which increases surface area for absorption. Yes, the root is going to absorb the water, but there are some extensions on the roots which make it even easier to absorb water. We went ahead and talked about guard cells, and we said that these ones are found in the leaves mostly, where we have the stomata. So the gut cells, we said, are specialized in such a manner that they have chlorophyll, and this chlorophyll is stored in structures known as chloroplast, as you can see on your screen. And the reason why these gut cells have a chloroplast is so that they can use the chlorophyll in the chloroplast to trap sunlight for photosynthesis. So when they carry out photosynthesis, it enables them to open the stomata, as we're going to discuss later in Form 2 work. We also said that in gut cells, the inner walls are thicker than the outer walls. And the reason why the inner walls are thicker is so that they do not stretch as much compared to the outer walls, causing the stomata to open easily. So we also went ahead and talked about specialized cell, palisade cell, which we said it is found on mostly on the upper epidermis of the leaf. And as you can see, this palisade cell has a lot of chloroplasts, which are used for the process of photosynthesis. Okay, so a leaf, especially green leaves, they have a lot of chloroplast in them, and the chloroplasts are packed in the palisade cell so that they can trap sunlight for photosynthesis. The principle here is the palisade cell is tucked on the upper surface, the surface of the leaf where the sun is in contact with the leaf. And because we have many chloroplasts on that surface, we are going to have high rates of photosynthesis taking place. So the plant is going to make its own food easily. So we also went ahead and talked about other specialized cells in animals now. And we said one of the specialized cell is a nerve cell. And we said that the function of a nerve cell is that it is used in transmission of impulses. And I remember we said impulses are they are electrical signals which are generated in the body and are transmitted along the nerve impulses. So the reason why you are able to feel pain when you get injured or you get pricked or burnt is because what has burnt you, we refer to it as a stimuli. It triggers your body to make electric signals, which we call impulses, and they'll be transmitted in your body, causing you to respond positively or negatively to whatever is causing the irritation. So we also went ahead and talked about the sperm cell, which we said its main function is to swim to reach the egg for fertilization. And we said it is specialized in such a manner that it has a tail, it has a head, which has nucleus that controls its activity and many other activities. So all those are examples of specialized cells. And we said that when we have several cells that are performing a similar function, are performing functions geared to achieving the similar goal, then we refer to them as tissues. So they are we have cells. When several cells perform a similar function, they become tissues. Like for example, red blood cells, white blood cells, and platelets. They may not perform the specific function that is very similar per se, but they work together for a specific reason, which is there a similar reason. That is, like for example, they are found in blood and they work all together in blood. So therefore, blood becomes a tissue because it has many cells in it. And then several tissues are going to be interconnected to form what we call organ. And then organs will form organ systems like the breathing system, the circulatory system, and then those organ systems will make an organism. So today, I want us to talk about cell physiology. And we said that, we talked about cell physiology, and we said that cell physiology is the study of functions of cells and how they relate. We also went ahead and talked about the cell membrane, where we say that the cell membrane is responsible for enclosing the contents of the cell, and at the same time, it regulates or controls what gets in and out of the cell because of the poles. We talked about the structure of a cell membrane, where we said it is made up of 2 layers of phospholipids and 2 protein layers. Now, there's an example I will give you as an assignment, and majorly it will compose of this subtopic, and also give you some questions in the group, which I'm expecting at least you have gone through those questions. And if you have a challenge in any of those questions, you can always feel free to inbox me or talk to me so that I can address some of the challenges that you have in that subtopic. So we went ahead and we also talked about properties of a cell membrane, where we said it is semi-permeable, it is sensitive to changes in temperature. We gave the importances for all those adaptations of the cell membrane. And then we also went ahead and talked about diffusion, where we said that physiological processes include diffusion, osmosis, and active transport. We discussed diffusion, all of it, and we said it's the movement of particles or molecules from a region of high concentration to a region of low concentration. We went ahead and talked about a practical activity where we can use potassium permanganate 7, which we said is purple. So when you place some particles of potassium permanganate 7 in a beaker containing clean water, you'll find that at the end of the experiment, we are going to have the particles of potassium permanganate 7 spreading across the beaker, because they'll move from where they are highly concentrated to where they are lowly concentrated. So we also went ahead and said we can practice or we can demonstrate diffusion using cologne or perfume, whereby the person who has worn the cologne will be highly concentrated with the particles of the cologne, but after some time, everyone else in the room will start smelling the cologne, because the particles will be moving from where they are highly concentrated to where they are lowly concentrated. So we went ahead and we also talked about how we can demonstrate diffusion using a whisking tubing. And you said that this is one of the experiments which we are going to do when we have time. Now today, I want us to begin the lesson by talking about factors affecting the rate of diffusion. Factors affecting the rate of diffusion. That's why we're going to begin our lesson today. Now, you see, we said diffusion is the movement of molecules or particles from a region of low concentration to a region of from a region of high concentration, to a region of low concentration. So a question comes in. Do these particles move at the same rate all the time? And do these particles have other factors, or do they have factors which can accelerate their movement or can slow down their movement? One thing we have to agree on is that particles move from where they are many to where they are few. So our question is, do they move at the same rate regardless of the changes in the environment? Or do we have some factors which can speed up the movement of particles or slow down the movement of particles? The answer to that question is yes, there are factors which might increase how fast these particles move or can even slow them down. So the first factor that affects the rate of diffusion is temperature. It is temperature. So when it is very, very hot, particles tend to move faster compared to when it is hot. So that means if you wear cologne or you spray your clothes or your skin with a powerful cologne on a very hot day, during around midday when it is very hot, people in that room will tend to smell that cologne faster compared to when you wear a cologne or you spray a cologne on your clothes on a very cold day. How is this possible? This is because when the temperatures are high, the kinetic energy, I want you to underline the term kinetic energy, okay? So energy that is causing the movement of molecules, okay? So if, for example, you're talking about molecules or particles of a cologne, they tend to move faster. Why? Because high temperatures increases the kinetic energy of molecules, causing them to move faster. So this explains the reason why in a room setup, when someone removes their clothes in their shoe, one of the shoe, and probably they have a smelly feet, alafu watoe kiatu in the afternoon, everyone in the room tends to smell that more faster or quickly compared to early in the mornings when one removes a pair of shoe if they have a smelly feet. Why is that possible? Because when it is very hot, temperatures increase the movement of molecules or particles, and this causes them to move faster. So therefore, how do you discuss that in an exam? If you're told to discuss factors affecting the rate of diffusion, you say, high temperatures increase the kinetic energy of molecules, causing them to move faster compared to low temperatures, which do not have the same effect. So in an exam, you'll say, temperature. High temperatures increase the kinetic energy of molecules. Therefore, the higher the temperature, the higher the rate of diffusion. I'm repeating. High temperatures increase the kinetic energy of molecules, causing them to move faster. Therefore, the higher the temperature, the higher the rate of diffusion. So that is one factor which affects or which is going to increase the movement of molecules. Another factor that is going to affect the movement of molecules is concentration or diffusion gradient. Concentration or diffusion gradient. Now I want to demonstrate concentration gradient or diffusion gradient. Now we are going to have two regions. We are going to have region A and we are going to have region B. So in region A, we are going to have very many molecules of a particular I hope it is visible. So we have very many molecules of a particular substance. And in region B, we have very few, like 5 of them. So in these two cases, region B is highly concentrated compared to region A. So region B is highly concentrated, region A is lowly concentrated. In this case, molecules are expected to move from region B to region A. Why is that possible? Because we said diffusion is movement of molecules or particles from a region of high concentration to a region of low concentration. So which is the region of high concentration? Region B. Which is the region of low concentration? Region A. So diffusion dictates that molecules should move from region B to region A. Now what is concentration or diffusion gradient? Diffusion gradient or concentration gradient is the difference in concentration between these two regions. Like for example, if you were to count the amount of molecules in region B, maybe they are 40 molecules. In region A, maybe they are 5. So what is the difference in concentration? The difference in concentration is 35. So that means the diffusion gradient or the concentration gradient is 35. So therefore, in any case where diffusion is taking place, if the concentration gradient is high, the rate of diffusion is high. So for example, again, if we have regions X and Y, and in region X we have 4 molecules, and in region Y we have 3 molecules, so the concentration gradient is just 1 because it is 4 minus 3. So you expect that the rate of diffusion of the molecule from region X to Y will be lower compared to diffusion between region B and region A. Why? Because there's a higher difference in concentration between region B and region A. So therefore, we can conclude and say the higher the diffusion gradient or the higher the difference in concentration between two regions, the higher the rate of diffusion. And that is how concentration or diffusion gradient affects the rate of diffusion. So first of all, the first thing nimesema, understand what is diffusion or concentration gradient. And this is the difference between two regions in terms of concentration. So the higher the difference, the higher the rate of diffusion. The lower the difference in the amount of substances or molecules, the lower the rate of diffusion. And that is how diffusion gradient or concentration gradient affects the rate of diffusion. So I hope that is well understood. Kindly, if you have understood that, type understood. Some of these concepts, it's only better you move forward if you are sure that the learners have understood. So those who are online, kindly type understood so that I can move to the other factor. I want to make sure you have understood that factor. Kindly just type understood so that we move on to the next factor. I'm waiting for the feedback kindly.
Understood, teacher.
Teacher, may I have a request?
Very good. I've seen the response
Kindly,
and the answer is you have understood.
teacher, may I
Let
have
us
a
move on.
request?
The
Teacher,
other factor which affects the rate of diffusion is size of the molecule. Size of the molecule. Now, I want us to consider molecules of bigger sizes and molecules of smaller sizes. Okay, so if I was to illustrate, let me give you an illustration of two molecules. Molecule A, which I'm going to draw in blue, that is the size of molecule A. That is molecule A. It is that big. And then we are going, so that is molecule A. And then we are going to draw molecule B, and that is the size of molecule B. So that is molecule B. Of these two molecules, which one is bigger? Of course, molecule A is bigger than molecule B. So how will this affect the way the molecules move? So of course, because A is bigger, you expect it to move more slower compared to molecule B. So since molecule B is small, it will move faster compared to molecule A. So therefore, when diffusion is taking place and it is involving molecules that are big, it means they will move slower, and therefore the rate of diffusion, the rate through which molecules will move from a region of high concentration to a region of low concentration, will be slower. So the larger the molecule, the lower the rate of diffusion, and the smaller the molecules, the higher the rate of diffusion. So if a molecule is bigger, it will move slower, slowly compared to a smaller molecule, and therefore the larger the molecule, the slower the movement of the molecule. So therefore the rate of diffusion will be low. The smaller the molecules, that means the molecules or particles will move faster, therefore the rate of diffusion will be higher. So I hope that is well understood. So in an exam, how do you discuss size of the molecules? You say smaller molecules diffuse faster compared to larger or heavy molecules. Good, that is it. So from there, we talk about another factor that affects the rate of diffusion, and that is surface area to volume ratio. Surface area to volume ratio. So therefore, the higher the surface area to volume ratio, the faster the rate of diffusion. The higher the surface area to volume ratio, the faster the rate of diffusion. Now we have another factor, factor number 5, which is thickness of the membrane. Thickness of the membrane. Now let me explain thickness of the membrane. So I want to draw two membranes here. The first membrane is, let me draw in red. The first membrane is membrane A, and then we have membrane B, which is not as thick. So in these two membranes, membrane A is thicker, meaning there's a longer distance from one end of point A to the other end of point A. While in membrane B, the distance is shorter. So therefore we say of the two membranes, membrane A is thick and membrane B is thin. So how will this affect movement of molecules? So if molecules are moving across two membranes, one which is thicker and the other which is thinner, as you can see, it will take more time for a molecule to move from this part to the other side of the membrane. Why? Because the distance it is going to cover is longer. Whereas in B, movement of molecules from this part here, that one, to the other part of the membrane B, it will take a shorter time because the membrane is thinner. So therefore you say, if molecules are moving across longer distances because of the thickness of the membrane, the molecules will diffuse slower. While if the membrane is thin, the molecules will diffuse faster. So how do you explain that in an exam? You say the thicker the membrane, the lower the rate of diffusion, while the thinner the membrane, the higher the rate of diffusion. Why? Because thinner membranes have shorter distances, while thicker membranes have longer distances to be covered by molecules. I hope that is well understood. And let's move on to the next factor affecting or influencing diffusion. So this factor is the type of the medium. That factor there, the type of the medium. Now, from science, we understand that we have three types of medium. The first type of medium is gas. The other one is liquid. Okay, and of course we have solid. But in this case, we are going to consider two mediums. The first one is gas and the second one is liquid. So between gases and liquids, where does diffusion take place faster? Of course, it is in gases. That is why mtu akikuja amevalia cologne, you will actually smell the cologne faster compared to maybe, for example, if that cologne has been put in water, for it to be dispersed across the water, it will take a longer distance. I mean, not distance, sorry, a longer time. So therefore, if diffusion is taking place across, if it is taking place across a liquid medium, it will be slower compared to across a gaseous medium. So we say in an exam we discuss and say diffusion is faster in gaseous medium compared to in liquid medium. Now in an exam, they have a funny way of testing this content. Number one, they can just ask you, list 3 or list 4 factors that affect the rate of diffusion. If they do that, you'll just say type of medium, thickness of the membrane, surface area to volume ratio, size of the molecules, temperature, and concentration or diffusion gradient. However, in other exams, they might require you to discuss those factors, like explain how temperature influences diffusion. You should be able to discuss and say, high temperatures increase the kinetic energy, therefore increasing the rate of diffusion. Concentration gradient, you say, this is the difference in concentration between two regions, and the higher the concentration, the higher the rate of diffusion. So therefore, those are two main ways. However, they have another way of testing this question that usually gets students off guard, which is that way. In an exam, you'll hear them ask you, or you read a question that is asking you to list 3 factors that increase the rate of diffusion. So in this case, they have tied you. You can only mention factors that increase. You cannot mention factors that decrease. You can only mention factors that increase the rate of diffusion. So for example, let's assume you're in an example and you have read a question asking you, discuss factors that increase the rate of diffusion. So in this case, you can only mention a factor that increases. For example, you cannot say temperature. Why can't you say temperature? Because temperature has two sides. When the temperature is low, the rate of diffusion is slower. When the temperatures are high, the rate of diffusion is faster. So how should you answer that question? You should say high temperatures. I'm repeating. If you get a question asking you to discuss factors that increase the rate of diffusion or increase the rate at which molecules or particles move from one region to another, you say high temperature. Because if you say temperature, in temperature it can either increase or decrease. But when you've been compounded to either saying how it increases, you can only say high temperatures. Another factor is concentration gradient. You cannot give an answer as concentration gradient. So how do you answer that question? You say high concentration gradient, because it is high concentration gradient that increases the rate of diffusion. Yes, it's true, size of the molecule affects the rate of diffusion. But what exactly in size of the molecules increases the rate of diffusion? You're going to say smaller molecules, movement on smaller molecules. So you'll say when smaller molecules are moving across from one region to another, they tend to move faster. Therefore, that increases the rate of diffusion. So smaller molecules. Then we have thickness of the membrane. So you'll say thin membrane. Thin membrane increases the rate of diffusion, and then type of medium, you'll say diffusion in gaseous medium. Diffusion in gaseous medium. That is going to increase the rate of diffusion. So when you're answering that question, read the question even twice to understand whether it is asking you to discuss, whether it is asking you to state, or whether it is asking you to name or state factors that only increase the rate of diffusion. I hope that is well understood, and if it is, we can comfortably move. Actually, you can write that question, write it down. Write it down so that you can answer it later. The question is, state 3 factors that increase the rate of diffusion. State 3 factors that increase the rate of diffusion. State 3 factors that increase the rate of diffusion. And those factors are the factors which we have mentioned there in the last like 5 or 10 seconds. Very good. I think we can comfortably move to the next concept because we have talked about factors affecting the rate of diffusion. Now that we have seen what increases the rate at which molecules move or decreases the rate at which molecules move, the right question to ask ourselves here, do we have roles of diffusion in living organisms? Or is diffusion essential or important in living organisms? Is there anything that diffusion can assist or can benefit living organisms? In this case, remember, living organisms includes animals and plants. Therefore, if we have benefits of diffusion in plants, we must also have benefits of diffusion in animals. So which are these benefits, or how is diffusion important? So that we say importance or role of diffusion in living organisms. Let's talk about diffusion in animals, benefits of diffusion in animals. The first benefit is diffusion is used in excretion. It is used in excretion or removal of waste from the organisms, most of the organisms. So diffusion is used in excretion or removal of waste from bodies of organisms. Then we have the other importance of diffusion in living organisms, which is gaseous exchange in respiratory surfaces. Gaseous exchange in respiratory surfaces. What does that mean? It means that how we carry out gaseous exchange. First of all, let's discuss what is gaseous exchange before I even explain that point. You see, you breathe in, right? Yes, you do. So when you breathe in, which gas do you breathe in? Which gas do you breathe in when you breathe in? Kindly,
Oxygen.
anyone?
Oxygen.
Anyone who will tell us the gas that you breathe in as an animal?
Oxygen.
This is easy. This is simple. Yes, Monica?
Oxygen, sir.
Anyone? Very good. When we breathe in, we breathe oxygen. I've seen an engagement in the chat box. So when we breathe in oxygen. It's okay. It's okay. So Mora is saying that I can't hear you. It's true. I didn't hear what Monica said, but at least you can type. That's good. That's okay. So we've said that when you breathe in oxygen, right? When you breathe out carbon dioxide. So the question is, what happens inside our body such that the body is able to differentiate between what we have taken in and what should come out? That is referred to as gaseous exchange. Now you see, our lungs are responsible for gaseous exchange. When you take in oxygen, that oxygen must be transported by the blood to all parts of the body. And when it moves to all parts of the body, it is responsible for one major function. And that function is respiration. What is respiration? Respiration is the process through which whichever food you have taken the whole day or previously or even 2 years ago or 3 years ago that has been stored in your body, that food is not stored for fun. It is stored so that one day it will be broken down by the cells of your body to give you energy for all the activities that you do. Energy to move from one place to another, energy to think, energy to transmit impulses, energy to jump up and down, energy to reproduce, energy to do a lot of things. Okay? When you take food, that food is going to be used for that process of respiration. How is it broken down? Of course, it is broken down in the cells in presence of oxygen. Are we together? So oxygen lazima ingie kwa mwili ndio itumike for respiration. I hope that is well understood. So therefore what it means is the body must take in oxygen. If it does not take in oxygen, you are not going to be able to break down food. If you cannot break down food, it means that you're going to have a problem and probably you will not even survive. When energy has been gotten from the body, from the food which you have eaten through respiration, there are products which come from that process. One of the products that is produced in that process is carbon dioxide. Carbon dioxide is a waste material. It cannot be used in the body in any way. So what that means is when you produce carbon dioxide, you are going to have to get rid of that carbon dioxide. And when you get rid of carbon dioxide, lazima itokee kwa lungs through the process known as gaseous exchange. Now, so that means in your lung, there must be an exchange of oxygen iingie kwa alafu carbon dioxide itoke. Okay? Oxygen iingie kwa mwili alafu carbon dioxide itoke. That process of exchanging carbon dioxide and oxygen is known as gaseous exchange. And how do those gases move in and out? They move through the process of diffusion. Kwa sababu ile hewa ina breathe in imejaa oxygen. So that oxygen is going to move from the lungs into the bloodstream because it is highly concentrated in the lungs and not in the bloodstream. What is highly concentrated in the bloodstream? Carbon dioxide, because blood is coming from all parts of the body. So this blood that is flowing to the lungs has a lot of carbon dioxide. So since carbon dioxide is highly concentrated in the blood, it will move from the blood into the lungs. Okay, so therefore diffusion takes place there, allowing carbon dioxide to move from the blood to the lungs and then oxygen from the lungs to the blood, and that is gaseous exchange being assisted or enforced by diffusion. So therefore you can say in animals diffusion is used in gaseous exchange. So I hope that makes a lot of sense and you have understood how. So this is not only in animals, it can also take place in gills. Okay, so like in fish, water that flows over the gill, you know the gaseous exchange structure in fish is known as a gill. So water that flows over the gill has a lot of oxygen, but inside the gills we have a lot of carbon dioxide. Where did the carbon dioxide come from? Carbon dioxide came from the respiration process in the tissues of the fish. So that one, because it must be excreted, it diffuses from the gills to the water that is flowing over the gills. And then, the water that is flowing over the gills has a lot of oxygen. So oxygen will move from a region of high concentration, which is in the water, into the gills. So therefore, also gaseous exchange will take place there. So in most of the animals, gaseous exchange takes place through diffusion because one gas is highly concentrated in one side, it moves to the other side. Then the other gas is concentrated from that other side and moves to the other side, causing movement of molecules or particles of that particular gas. And that is how gaseous exchange is important in animals. Gaseous exchange is the second role or importance of diffusion. Then we have absorption of digested food. Like for example, when you take any food, the first thing that happens is you chew the food to increase surface area for digestion. When you digest that food, it moves to a very important part of the human body of the animal, which is the small intestine. In the small intestine, you're going to have food materials being absorbed by the gills. Not the gills, sorry, by the by the small intestine into the bloodstream. Okay, so when that happens, remember in the blood we do not have food materials, we just have blood flowing in the blood vessels. Okay, and then inside the small intestine we have a lot of food materials, like for example glucose, we have also sucrose, we have for example glycerol. So we have every food material that has been digested in all the digestive system, and it is in the small intestine. So those materials, because they are highly concentrated in the small intestine, they will move from the small intestine and diffuse across the small intestine into the bloodstream. That movement of food materials from the small intestine into the bloodstream is going to be facilitated by diffusion. I hope I'm not very fast. I'm saying once you digest food in your blood, in your body, through the digestive system, from chewing, swallowing into the stomach, we're going to see that process. Proteins get digested in the stomach, in the duodenum, some more food materials will be digested. Even in the small intestine, some food materials will be digested. The smallest forms of those food that has been digested, they will be absorbed in the small intestine through the process of diffusion. How is that possible? Because in the small intestine we have a lot of food materials, but in the bloodstream we do not have food materials. So therefore materials will move from where they are highly concentrated, which is in the small intestine, into the bloodstream. I hope that is well understood. So therefore we can say that diffusion is important, for it allows absorption of food materials. Okay. Point number 4 on importance of diffusion in animals is that diffusion is responsible for reabsorption of useful materials and some salts in the kidney. So when we will be doing Form 2 work, we're going to talk about the kidney, and we will see how some food materials get reabsorbed. So for now, I want you to master these 3 roles of diffusion. Role number 1, excretion of waste materials. Role number 2, gaseous exchange. Role number 3, absorption of digested food in the small intestine, or what we call the ileum, into the blood stream. That is when it comes to animals. So when it comes to plants, diffusion also plays a very important role. So one of the importances of diffusion in plants ni number 1, absorption of mineral salts by plant roots. So in the roots, in the soil, sorry, we have some nutrients which are very important for plants. So those nutrients are going to be absorbed through the process of diffusion. So, like I'm saying, that once the plant wants to absorb materials, those materials can move from the soil to the roots to be taken up the plant, and this is going to take place either through diffusion or another process which we call active transport. But we're going to talk about active transport. For now, what I want you to understand is that diffusion is responsible for absorption of mineral salts from the soil into the roots. And when these mineral salts are taken into the plant, they are used in the process of making substances that are useful in the plant. So that is another way in which diffusion is used in plants. A second way in which diffusion is important in plants is it helps in gaseous exchange. It helps in gaseous exchange. Just like I have explained, nimesema two gases which are used in gaseous exchange are carbon dioxide and oxygen. They are carbon dioxide and oxygen. So what happens is the difference between gaseous exchange in plants and animals is in animals they take in oxygen, they give out carbon dioxide. So oxygen taken in will be used in respiration and carbon dioxide will be obtained from the respiration process. Then we have in plants. In plants, they will take in carbon dioxide and they will remove or they will excrete oxygen. How is that possible? It is very possible. Do you know how it is possible? Plants take in carbon dioxide and break it down and combine it with hydrogen from the water. We're going to see that process, okay? But then, one of the products that come from the process of photosynthesis is oxygen. And plants release that oxygen to the environment, and then that oxygen will be absorbed by animals. So therefore, exchange of carbon dioxide and oxygen in plants also takes place through diffusion. Okay? So therefore, we can comfortably say gaseous exchange in plants also takes place through diffusion. I hope I'm well understood. And finally, the last importance of diffusion in plants is that it is important in transport or translocation of manufactured food from the leaves to other parts of the plant. Since the leaves are the ones that carry out photosynthesis, the food material now can move freely from the leaves to all parts of the plant through diffusion. How is that so? Because in the leaves there is high concentration of food material, while in other parts of the plant there isn't, so materials will move easily from where they are highly concentrated to a region where they are lowly concentrated. Bottom line here is understand what diffusion is. Know that diffusion is the movement of materials or molecules from a region of high concentration to a region of low concentration. So the next question you ask yourself is, how do they move? They move from where they are highly concentrated to where they are lowly concentrated. So which factors affect movement of these molecules? Do they move faster when it is hot or when it is cold? So that's a factor. So when it is hot, they move faster. When it is cold, they move slower. And then you ask yourself, what do we call the difference between two regions? Region A and region B. That one we refer to concentration gradient or diffusion gradient. So that means if there is a greater difference between region A and region B or whichever two regions, that will increase the rate of diffusion. And if there is low difference in concentration, then that means the rate of diffusion will be lower. So if we have a thicker membrane, it will take longer for molecules to move, and therefore the rate of diffusion will be slower compared to when we have a very thin membrane where diffusion rate is going to, of course, be higher. And then we also have the type of medium, so such that when we have a higher medium, a gaseous medium, the rate of diffusion will take place more compared to when we have a liquid medium. Then we have roles or importances of diffusion in living organisms, and we have divided that into two, that is in animals and in plants. Something that I also want you to note here is there are some similarities between plants and animals in terms of roles of diffusion. Like, for example, we have gaseous exchange. So when you look at that, we have gaseous exchange in plants and gaseous exchange in animals that are similar. So in an exam, there are two ways of testing this content. Number one, they can ask you, state three roles of diffusion in plants. Or they can ask you to state 2 or 3 roles of diffusion in animals. Or the second way of testing this area needs, they can ask you a general question. List or state 3 roles of state 2 or 3 roles of diffusion in living organisms. state 2 or 3 roles of diffusion in living organisms. So when they say that, you should not differentiate. You should just give either whether ni a plant ama ni a animal. But wakijaribu wakwambie you state the role of diffusion in plants, you must give the ones which are exclusive, only taking place in plants. And if they ask you to give the roles of diffusion in animals, you must only state the ones which give the specific role, the specific roles of diffusion in animals and not in plants. So that is diffusion in a nutshell. And of course, there are some several questions which we can do, like these ones which you calling study questions. So if we are comfortable, I think I can display some questions for you, and then we can do some questions on diffusion before we move on to something else. So if you're in favor of doing several revision questions on diffusion, kindly raise up your hand so that we can do 2 or 3 questions on diffusion before we move on to osmosis. So raise up your hand so that we can do. Tell me you're together with me so that we can do several questions on diffusion, kindly. Good. So I think we are all in favor of doing several questions. So give me a minute so that I share. Good. There you go. So we have some questions that I'm sharing there on the role of diffusion or on diffusion in general. We are going to begin with the first question, which is, I want us to do just questions on diffusion. And I'm sure I gave you these questions, so I'm sure you have done some of these questions. So question 2A. Question 2A is, define the following terms. The first term is diffusion. Question 2A, define the following terms. The first term is diffusion. So is there anyone who is willing to define diffusion for us? Anyone who is in the lesson today and is willing to define diffusion for us?
Can I try? Yeah, diffusion. Diffusion is the movement of particles from higher concentration to low concentration.
I want to believe that I want to believe that Domintila. I hope I got that right. I want to believe you were saying something, but mimics just hear anything. Is there anyone in the group that can hear? It looks like, I hope you can hear me. I hope ninyi mnanisikia, because from my end, siwasikii.
But mwalimu, si tunakusikia. Hujanisikia bado.
Sasa ni wewe nimekusikia, Monica nimekusikia. So do you want to define what is diffusion for us?
Yes. I said it is the movement of particles. Yeah, it is the movement of particles from higher concentration to low concentration.
Very good. That is correct. That is correct. Good. So diffusion is movement of molecules or particles from a region of high concentration to a region of low concentration. That is correct. Thank you for that. Let's look for another question on diffusion. We have question 4. I hope we are all there. So question 4. Sorry? Yes, good. So question 4. Let's do question 4 together. The setup below illustrates a certain physiological process. Okay, so when you look at that setup, we have molecules of dye there on one side, then we have water, and across we have a membrane. And then of course we have the first one. The second one, if you check well, you will see we have some molecules which are moving from the first region where we have the dye to a region where we have said we have water. So ukiangalia vizuri utaona hapa kwa maji, at the beginning we didn't have any molecule of the dye, but in the second setup, you can see very well that the number of molecules have increased. So hizo kuko na 1 2 3 4, so we have 4 molecules. And then in the third setup where wameandika equilibrium, utaona the number of molecules on that side and the number of molecules on the other side are equal. So what that means is there was movement of materials, and which materials moved? The dye molecules. So that is the observation that we have made. So I think therefore we can comfortably do the questions. First question, name the physiological process that is being illustrated. Name the physiological process that is being illustrated. So in this question, the first thing you should do is check to see what is moving. The first thing is to check what is moving. And in this case, it is the dye molecules that are moving. So therefore, what is the answer now? The molecules have moved from hapa ni mingi, huko hakuna. At the end of the day, zote zimetoshana. So what is the answer to that question? The physiological process is, of course, diffusion. Very good. Very good. That is correct. It is diffusion. Even the ones who are typing, nimeona mmepata correct. Mmepata hiyo answer ikiwa correct. So the physiological process is diffusion. Okay? Higher. We go to question 4b. Give 3 examples of the process named named above in plants. Give 3 examples of the process named in A above in plants. So in this case, they have been very, very specific. They did not tell you in living organisms. They did not tell you in animals. They told you in plants. So in short, what they're asking here is state 3 roles. Ukiona wamekwambia list 3 or give 3 examples of the above named process. This means you must give the roles. Hapa unaulizwa roles. Na sisi tumezisoma. So the first role is anyone? The first role of diffusion, ama first way in which diffusion is useful.
Transport of manufactured
First way in
food.
which diffusion, sorry?
Transport of manufactured food.
Very good. Transport of manufactured foods from the leaves to all parts of the plant. That is very good. Another answer. We have been asked 3. So we've given 1. Another one. Sorry?
Absorption of mineral salt.
Sorry? Sijasikia hiyo. Mekatika kidogo.
Okay. I repeat. Absorption of mineral salts.
Very good. Absorption of mineral salts from the soil by the roots. Absorption of mineral salts in the soil from the soil by the roots. And finally, the last one.
Excretion of CO2.
Yes, gaseous exchange. Very good. Gaseous exchange. Okay. So exchange of carbon dioxide and oxygen in the stomata. So I repeat the answers that have been given and they are correct. The first one we've said it is gaseous exchange. The other one, absorption of mineral salts. And the other one, of course, we've said movement of food materials from the leaves. Now, this is one. If wangekuuliza, you state the activities in animals that are similar to that process or roles of diffusion in animals, which ones will you have given? If they replace now from plants to animals.
Excretion.
Excretion, very good. Another one.
Gaseous exchange in respiratory surfaces.
Sorry?
Gaseous exchange in respiratory surfaces.
Very good. Gaseous exchange in respiratory surfaces. Another one.
Absorption of digested food.
Yes, very good, very good, very good. Yes, that is well understood and stated. Absorption of digested food in the small intestine. I hope mnaona venye najibu swali. Nasema absorption of food, na sijamalizia hapo, nimesema in the small intestine. So gaseous exchange in respiratory surfaces. So that is the correct way of answering such a question, because it shows you know what you're talking about, and it gives you, it shows that you have understood, we have a greater understanding of that concept. So it is good to answer a question that way. So from there we can move on to the next question, question C. Now state two ways by which the movement of the dye in the setup would be slowed down. A very good question. State two ways by which the movement of the dye in the setup would be slowed down. Now remember tulisema, movement of molecules from a region of high concentration to a region of low concentration is what we define as diffusion. And then we said molecules can either move faster from one region to another or slowly, depending on some factors that we mentioned. Those factors, let me remind you, tulisema ni temperature, thickness of the membrane, tulisema surface area to volume ratio, tukasema the medium, tukasema all those factors. So the question is asking you, state two ways by which movement of the dye in the setup, si ati it would be increased, no, tumesema slowed down. So anyone who will tell us ways in which we can slow down the movement of the dye? Anyone?
Reducing the temperature.
Very good. Reducing the temperature. Because when we increase the temperature, we will increase the rate of movement. So slowing down involves reducing the temperature. Very good. Another way?
Increasing the thickness of the membrane.
Very good. You see that membrane that they have drawn there? If you increase the thickness of that membrane, we are going to have molecules of the dye moving across longer distances, and this will slow down the movement. I'm very happy. I'm very happy. Another answer? Another way?
Increasing the sizes of the molecules.
Very good. You can increase the size of the molecule so that you can have very bigger molecules which will move slowly in that particular setup. I'm very happy with the engagement, and I'm also very happy to understand that we have understood the concept. So
teacher,
that is how you should have answered that question. A question?
Yeah, teacher.
Mm-hmm.
Can adding more water slow down the movement of the dye?
No, it will not slow down because if you add more water, water is still the medium. Okay, so in this case, adding more water will not cause the slow movement, because at the end of the day, water is still water. Okay, so not unless now we talk about changing the medium. But of course, we said the two mediums are gases, and we have, of course, liquid. And in gases, now when we change the medium into gaseous medium, this will increase. So in this case, adding more water will not reduce the almost slow down the movement of particles, because at the end of the day, it is moving in the same medium, which is water. So the rate of movement will still remain to be the same. So is that understood?
Yes, very well.
Very good. So we can now move on to the next question. Let us move on to question number 5. Question number 5. So I'll read the question for you. Now remember, in an exam, before I read question number 5, hapa question 4C, they can still ask you now the opposite. Instead of slowing down, wakuulize how to increase or to speed up. So you should also be able to answer such questions. Actually, so we can answer that. So let me reread the question C, but now rephrase it. State two ways by which the movement of the dye in the setup would be increased. So we're answering that question. How will the movement of the dye be increased?
Increasing temperature.
Increasing the temperature.
Yes, increasing the temperature. Good. Go ahead.
Reducing the thickness of the membrane.
Yes, reducing the thickness of the membrane. Good.
Reducing the size of the molecules.
Yes, reducing the size of the molecules of the dye.
Increasing the concentration gradient.
Very good. Increasing the concentration gradient. Nyinyi ni wanafunzi wazuri. I'm very happy. So we do question 5 and then I will allow you to do other questions by yourselves so that we can talk about osmosis for a moment. Okay? So is that in order? We do question 5 and then we move on to osmosis.
Okay, yeah.
Okay, good. Question 5. A group of students set up an experiment to demonstrate a certain process. The experiment was set up as shown in the diagram below. Now I want you to be very keen with this question because this question can be set up in diffusion and also we can have a similar setup in osmosis. The only difference is the contents of what is inside the beaker and inside the whisking tubing. First of all, before we even do the question, I want us to agree on what a whisking tubing is. And during the previous lesson where we were talking about the cell membrane, we said you can have a synthetically made structure that will function similar to a cell membrane, and that structure is known as a whisking tubing. Now, a whisking tubing basically functions like a cell membrane. So it allows materials to move in and out from what is inside and what is outside. So the reason why I'm saying that we need to concentrate on this setup is I want us to identify the content. So before we even answer the question, at the beginning of the experiment, I want us to look at that beaker. In that beaker, we have that brownish color liquid, that brownish liquid, that brown liquid. The name of that brown liquid is iodine. It is iodine. And then we have inside the whisking tubing a white solution. That white solution is referred to as a whisking tubing. So we have iodine solution and we have starch solution inside the whisking tubing. Now, I want you to also move your concentration or your attention to the end of the experiment. So when you move your attention to the end of the experiment, there's something you're going to note. You will note that inside the beaker, the solution's color remain the same. It is brown. Okay? But inside the whisking tubing, something has happened. The color has changed from white to blue color or what we say blue black. In biology we say blue black. So at the beginning of the experiment, inside the whisking tubing there is starch which is white in color. At the end of the experiment, the starch solution inside the whisking tubing changes color to blue black. Now this concept is very important. So every time you're doing a question that has any diagram, the most important thing is to study the diagram and understand. Now, this concept here is testing about a physiological process. And if you check very well, some movement has taken place. There are some materials which have moved, and by the look of things, it is probably iodine that has moved inside the starch solution. Now, there's something I want to bring to your attention, and this is something we call test for starch. In a laboratory, if you want to know whether a food substance has starch, we use iodine. How does it work? When you when you put several drops of iodine in a particular substance that contains starch, the color of iodine changes from brown to blue black. That is very important to note, that every time you want to test for starch, the best reagent to use in the lab is iodine solution. And when iodine solution comes into contact with starch, it changes the color of starch from white to blue black, or starch will change the color of iodine from brown to blue black. So when there's a contact between starch and iodine, the color changes automatically to blue black. So in this case, you can see the blue black color is not inside the iodine, it is inside the whisking tubing, meaning starch did not move from the beaker. I mean, from the whisking tubing, sorry. It moved, what moved is iodine from the beaker into the whisking tubing. If you look at the whisking tubing at the start of the experiment, it appears smaller compared to the whisking tubing at the end of the experiment, suggesting there are some iodine which have moved. Even if it's not a lot of iodine, so long as there's contact between iodine and starch, the color changes to blue-black. So in this case, if you study the diagram well, you will see the movement of materials was not from the whisking tubing to the beaker, but it was from the beaker into the whisking tubing. That is why the color of the iodine inside the whisking tubing has changed to blue-black. So in this case, diffusion took place moving from the beaker and the molecules moved into the whisking tubing. So in an exam, how do you know that the physiological process being investigated is diffusion? You check the contents of the beaker. Ukiona mahali wameandika iodine na kuko na starch solution, automatically that should tell you they are testing for for diffusion, because it is through diffusion that the iodine solution will move from the beaker into the whisking tubing. So with that understanding, I believe now we can go ahead and do the questions in that particular question. Higher, so after 10 minutes, the students recorded their observation in the table as shown below. So at the start of the experiment, inside the whisking tubing, the color was white, and outside the whisking tubing, the color was brown. At the end of the experiment, inside the whisking tubing, the color now changed from white to blue-black, while outside the whisking tubing, the color changed from brown to, it did not change, it was retained. The color was retained. It was brown at the beginning of the experiment, and it ended up being brown at the end of the experiment. So question A, name the physiological process being investigated. So the physiological process, of course, tumesema, the moment you see iodine and starch, automatically that one will have to become diffusion. So the answer to that question is diffusion. So the other question is, explain the results obtained in the setup. Explain the results obtained in the setup. So the first question is, which results were obtained? Which results were obtained? So the results that were obtained, anyone who will tell us the results that were obtained at the end of the experiment? Anyone? To explain the results that were obtained, actually just state what was observed at the end of the experiment. Anyone?
I can try, teacher.
Yes, go ahead.
The color of the whisking tubing changes to blue-black, while the solution in the beaker remained brown.
Very good. That is correct. That is what was observed. That the color of the whisking tubing changed to blue-black, while the iodine color in the in the beaker remained brown. But swali imekuambia explain the results. So when you're told to explain the results, you get these 3 marks by number 1, stating what was observed, and number 2, explaining. So in this case, you get 1 mark for stating that the color of the whisking tubing changed to blue-black, while the color of iodine remained brown. Why did that happen? Of course, it is because there was movement of materials. So you basically explain what moved from where to where and why. Anyone who will try? Okay, go ahead.
The walls of the whisking tubing is semi-permeable, and it will allow the iodine molecules to pass through from the beaker, and then the iodine will react with the starch, And then the color changes
Very
to
good.
black.
That is correct. Yeah. What do you think? Why do you think that starch molecules are not going to pass from the whisking tubing into the beaker? Why?
Because they are too large to move out.
Yes, that is very correct. So your answer is correct. So in an exam, you say that since the molecules of iodine are smaller, they move from the beaker into the whisking tubing, and this causes the color to change to blue-black, while starch molecules being bigger than the iodine molecules will not move from the beaker into from the whisking tubing into the beaker. So I repeat, I've said, since the molecules of iodine are smaller than those of starch, iodine molecules move into the whisking tubing by diffusion, while starch molecules remain in the whisking tubing. This causes the color change in the whisking tubing from white to blue, black. So that is the concept. And finally, explain the results expected if the experiment was repeated using starch solution, which has been boiled with dilute hydrochloric acid. A very good question. I repeat, explain the results expected if the experiment was repeated using starch solution, which has been boiled with dilute hydrochloric acid. I want to see if there's someone who is going to try and answer this question. Anyone? Okay. Go ahead.
Teacher.
Mhm.
I think the color inside the whisking tube will not change. And the HCl will kind of break down starch to sugars. Into simple sugars.
Into simple sugars, eh? That is very correct. That is very correct. That is correct. Now, let me explain a bit. So, I want us to talk about carbohydrates. Okay? Now, carbohydrates are divided into three categories. We have the first category, which is the simplest form of carbohydrate, which is monosaccharides. Monosaccharides are the simplest form of carbohydrates. When you combine two monosaccharides, you form a disaccharide. Disaccharide. Di means two. So you have one monosaccharide. An example of a monosaccharide is glucose. Okay? So the simplest form of a carbohydrate is a monosaccharide. For example, glucose. So when you combine two glucose molecules, you form a disaccharide. A disaccharide is made of two monosaccharides. A disaccharide is more complex compared to a monosaccharide. However, the most complex form of a carbohydrate is a polysaccharide. It is a polysaccharide. Now, polysaccharides are the complex form of carbohydrates that are formed by more than two monosaccharides. A very good example of a polysaccharide is starch. So therefore, starch is more complex compared to maltose and glucose. As a matter of fact, when plants carry out photosynthesis, they store carbohydrates in form of starch. So what it means is iodine can only react with starch. If you mix iodine with simple sugars like maltose and glucose, iodine's color will remain the same. Now, how can you break a polysaccharide such as starch into a monosaccharide or a disaccharide, or what we call simple sugars? Simple sugars means they are not as complex as starch. So one of the ways in which you can break down a complex starch into, a complex carbohydrate into a simple sugar is by number one, digestion. And that explains the reason why tunakulanga ugali, saa zingine tunakula githeri, saa zingine tunakula rice. So it is carbohydrates which are complex, that is starch. And then they get broken down through digestion, into simple sugars. Another way in a laboratory how you can break down complex carbohydrates into simpler sugars is by boiling them in presence of an acid such as HCl. So when you boil starch in presence of an acid such as HCl, that starch will be broken down into a simpler sugar or into a simpler carbohydrate such as maltose and glucose. So if you happen to boil starch in HCl, what is going to happen is the starch will now be converted into maltose and glucose, which are simpler carbohydrates. So therefore, even if iodine comes into contact with the simple sugars such as maltose and glucose, there will be no color change. Why? Because iodine only changes in presence of starch. So the answer here, just like we've been told, is that the color of iodine will not change because starch will be broken down by hydrochloric acid into simpler sugars. So there will be no color change of iodine because starch will be broken down into simpler sugars by HCl. So that is very correct, and I'm happy with the way you have contributed to today's lesson. I'm very, very happy. It shows we have understood. So today, for the last subtopic of the day, I want us to talk about osmosis. We are not going to strain with osmosis. We're just going to define what osmosis is. We're also going to see how osmosis is important in plants and animals. And then tomorrow, because we have a lesson in the morning, we will talk more about osmosis. So allow me to now switch from questions and start showing notes for us to be able to move on. So osmosis is the next subtopic that we are going to discuss for the day. Osmosis. Now, one thing I want you to note about osmosis is osmosis basically involves water. Maji peke yake. Okay? So if you do not see water, then that is not osmosis. If it does not involve water, then that is not osmosis. If it does not involve, I want you to always say that. If it does not involve water, it is not osmosis. It has to involve water to be osmosis. So therefore, what is osmosis? Osmosis is the movement of water molecules from a region of high concentration of water molecules. Let me pause there for a minute. Osmosis is the movement, I'm repeating, osmosis is the movement of water molecules from a region of high concentration of water molecules. What does that mean? It means that it is the water molecules that are moving. It is not anything else that's moving. It is the water molecules. Na ile place yenye kuko na maji mingi, we say that we have high concentration of water molecules in that region. So for example, tukiweza kuwa na maji fresh, hayo maji yako na molecules mingi sana ya maji, and therefore that is a highly concentrated region with water molecules. Okay? So when water molecules move from where they are high water molecule concentration to a region of low water molecule concentration through a semi-permeable membrane, we say that that is osmosis. Now let me explain something here for a second. So assuming we have two regions, tuko na region A. I like teaching these concepts in regions. So we have region A and we have region B. In region A, to assume we have sugar solution, you're going to assume we have sugar solution, And in this solution, we have 5 molecules of sugar. And region B, we have 10 molecules of sugar. So in region A, we have 5 molecules of sugar. In region B, we have 10 molecules of sugar. In this case, if we refer to water molecules, the region with more water molecules is region A. Why? Because sukari ni kidogo kushinda maji yenye iko huko. Okay? But in region B, we have 10 molecules of sugar, which means that there is more sugar in region B than water. So therefore, in these two regions, region A is highly concentrated with water molecules, while region B is highly concentrated with sugar molecules. So in this case, where will water molecules move from? Water molecules will move from where they are highly concentrated, which is in region A, to where they are lowly concentrated, which is region B. So basically, osmosis is the opposite of diffusion. So mahali kuna maji mingi, hapo maji huwa inatoka, inaenda kwenye kuko na maji kidogo, and that becomes osmosis. And then there's a condition for osmosis to take place. It must be across a semi-permeable membrane. Meaning, if we do not have a semi-permeable membrane separating two regions, hakuna osmosis ina take place. Not even a single. So there will be no water molecule moving if we do not have a cell membrane. So therefore we can say osmosis is the movement of water molecules from a region of high water molecule concentration to a region of low water molecule concentration across or through a semi-permeable membrane. So osmosis, I'm repeating for the last time, is the movement of molecules from a region, water molecules, movement of water molecules from a region of high water molecule concentration to a region of low water molecule concentration across a semi-permeable membrane. Now, what are the most important points to note? Usually we say osmosis is a special type of diffusion. Why do we say that osmosis is a special type of diffusion? Because number one, it allows movement of materials. But these materials that are moving are water molecules. So it is special in such a manner that it is water molecules that are moving. It is also special in a manner that it can only take place across a semi-permeable membrane. Despite the fact it is movements that are moving, these molecules must move. The molecules must move across a semi-permeable membrane. So therefore we say osmosis is a special type of diffusion for those reasons. If you look at the diagram that I have illustrated here, we have a region where we have distilled water and we have another region where we have sugar. So in this case, water molecules are moving from where there are many, that is in the distilled water region, and then those molecules are going to move across the semi-permeable membrane into the other part where we have sugar solution. So in these two cases, the region where we have sugar solution is highly concentrated with sugar, and the region where we have distilled water is where we have high water molecule concentration. So therefore, osmosis will cause the molecules to move from the region of high distilled water concentration to a region of high sugar molecule concentration. So if you're defining osmosis, you must mention three things actually. The first thing that you must mention is osmosis is movement of water molecules. You must also state that osmosis involves movement of water molecules from a region of high water molecule concentration to a region of low water molecule concentration. And then the last thing you must mention is it takes place across or through a cell membrane. It takes place across a cell membrane. Her terms used in osmosis. Before we even talk about terms used in osmosis, I want us to actually look at osmosis, and I want you to tell me whether you have understood what osmosis is. So if you have not understood what osmosis is, kindly type for me not understood so that I can repeat. If you have understood, then you can type for me understood, or you can ask any question that you have. The floor is open. Anyone with a question? Good. So if we have understood, like you have typed, then I think we are good to go. Now, before we talk about terms used in osmosis, let us talk about roles of osmosis in plants and animals. Roles of osmosis in plants and animals, okay? Roles of osmosis. It is very important. So how do animals use osmosis in their daily life? The first role of osmosis is that in plants, osmosis is used in absorption of water by the roots. In plants, osmosis is used in the absorption of water by the roots. So you find that the roots are going to use osmosis to absorb water from the soil. That is, of course, through the root hairs. The other role is that it helps in support in herbaceous plants. What are herbaceous plants? Herbaceous plants are non-woody plants. Like, for example, I'm sure that you guys have come across something like coriander, dania, okay? Dania does not have a woody stem. That is why it is easily chopped and cooked when you're cooking food. So a plant like dania, since it does not have a woody stem, that plant is going to use water for support. So when the cells of the coriander get hydrated with water, they become very stiff and they are used in the process of they are used to keep the plant standing in the environment, which causes the plants to carry out photosynthesis. It is exposed to sunlight for photosynthesis. So therefore there are some plants which do not have woody stems that depend on osmosis for support. That means that the cells will gain a lot of water and become turgid for the environment so that they can trap sunlight. Good. The other role of osmosis in plants is that it helps in movement of water from cell to cell. Water moves from one cell to another through a process known as osmosis. How is that possible? This is possible because one cell is separated from another cell using a cell membrane. So water can easily move from one cell to another because there's a cell membrane separating two cells, and therefore water molecules can easily move. And remember, we've said that osmosis involves movement of water molecules through or across a semi-permeable membrane. So therefore, when two cells are bordering one another, water can easily move from one cell to another because of the cell membrane, and this one will take place through osmosis. Okay. Another importance of osmosis in plants is that it helps in movement of water from cell to cell, and then the other one is it enables opening and closing of the stomata. Now we are going to discuss this further when we'll be talking about gases and explosive plants. But it is good to know that the movement of the opening and closing of the stomata in a plant is going to be affected by osmosis. How does that happen? When the guard cells, remember to say guard cells are specialized cells. So when the guard cells gain a lot of water from the neighboring cells, they swell, causing the stomata to open. At night, when the guard cell wants to close the stomata, the guard cell loses a lot of water to the environment, not to the environment, to the surrounding cells, and it is going to shrink, hence closing the stomata. We are going to discuss that and we'll see how that happens in Form 2 work. For now, just note that osmosis is used in opening and closing of the stomata. The other one is feeding in insectivorous plants. So osmosis is used in feeding in insectivorous plants. Now I want to give you an assignment. Today or tomorrow when you have time, and probably maybe today, okay, or even tomorrow, based on your pace. When you have time, I want you to search this in on YouTube, and I'll search insectivorous plants, or you can search feeding in insectivorous plants. Feeding in insectivorous plants. In insectivorous plants. You're going to understand how feeding takes place in insectivorous plants very easily. There are very good short videos on how that happens. And you'll also note the reason why insectivorous plants feed on insects is because they grow in soils that have less nitrogen. Okay, so those soils are very poor in nitrogen, so therefore the plants need to feed on insects. But for now, what is connected to osmosis is feeding in insectivorous plants, and I've given you an assignment. Kindly go check how insectivorous plants are going to feed using osmosis, and also I'm also giving you also another assignment. Go to YouTube and search opening and closing of the stomata, and you'll get some more explanatory videos. Sometimes you can even watch some animation videos. They are very good. They're animated videos on how the stomata closes and opens, and how insectivorous plants are able to feed. Okay, yeah, so when you get time, kindly check those two roles of osmosis: feeding in insectivorous plants and opening and closing of the stomata. So we said we have absorption of water from the roots, then tumesema support in herbaceous plants, then tumesema movement of water from one cell to another, and then enables opening and closing of the stomata, and finally we have said it helps in feeding in insectivorous plants. Let's talk about roles of osmosis in animals. Role of osmosis in animals. So the first role of osmosis in animals is that it enables reabsorption of water in the kidney. It enables reabsorption of water in the kidney. Role number 2, it helps in movement of water from cell to cell. It helps in movement of water from cell to cell. And role number 3 of osmosis in animals, it helps in absorption of water from the alimentary canal. So pale kwa large intestine, when you drink water, your body absorbs water from the large intestine, or what we call colon. So ukikunywa maji yako inaendaga kwa large intestine, hapo ndio maji huwa inakuwa absorbed. And the process by which water will be absorbed in the body, of course, it is through osmosis. So those are the roles of osmosis in plants and animals. So in an exam, you can get a question asking you that. But the highly examinable area here is roles of osmosis in plants. They are highly examinable, and the good thing is that they are many and they are easy to remember. Okay, so the last thing that I want us to talk about today is differences between osmosis and diffusion. Differences between osmosis and diffusion. So the first difference is diffusion involves movement of particles or molecules. It involves movement of particles or molecules, while osmosis involves movement of water molecules. Get that difference? Osmosis involves movement of water molecules, while diffusion involves movement of particles or molecules. Difference number 2. Diffusion takes place either through a membrane or even air. So diffusion can take place both kwa membrane and even kwa hewa. But for osmosis to take place, it must be through a semi-permeable membrane. Osmosis must only be through a semi-permeable membrane, meaning without a semi-permeable membrane, there is no osmosis. And then finally, the third difference is that diffusion is not affected by changes in pH, but osmosis is affected by changes in pH. Now, how is that possible? Remember pale kwa introduction tukisema kuhusu cell, tulisema that we have cell organelle, cell membrane, and we said that a cell membrane is semi-permeable. So an example of a semi-permeable membrane where osmosis takes place is a cell membrane. And we said some of the properties of cell membrane mean they are sensitive to changes in pH, meaning when a cell membrane or a cell is exposed to extreme pH, it gets denatured. Denaturing means it gets destroyed. And once a cell membrane has been destroyed, it does not allow movement of materials from one point to another. So therefore, because diffusion takes place even in air, it cannot be affected by changes in pH. But for plants, not plants, but for osmosis, because it must take place across a semi-permeable membrane, therefore, it means there must be an effect if in case the pH is altered or changed, because the pH will destroy the cell membrane, and then of course without the cell membrane, osmosis is not going to take place. So in an exam, if they ask you to give the differences between osmosis and diffusion, the easiest one is that diffusion involves movement of molecules and particles, while osmosis involves movement of water molecules. And then we've said diffusion can take place through a cell membrane or also through air, and then the osmosis only takes place through a semi-permeable membrane. And then finally the last one is that diffusion is not affected by changes in pH, while osmosis is affected by changes in pH because it only takes place across a semi-permeable membrane, which is sensitive to pH changes. So those are the differences between diffusion and osmosis and their roles. So when we meet tomorrow in the morning, we are going to talk about terms used in osmosis. Terms used in osmosis, so that we can understand some of these concepts, and then I'm sure we will be okay. So kesho tukipata chance ya kufanya hizo terms used in osmosis, on Saturday, because I've seen we have a lesson, we will majorly do questions on osmosis. We do like around 10 to 15 questions on osmosis, so that we can comfortably say we can move to another physiological process. So the remaining minutes, I'm giving you a chance to ask a question if there is any question. So I'm open to take questions, whether they are related to this concept or whether they are not. I will take the questions. Whether they are related or not, I will still take the questions. So the floor is yours to ask any question which you have come across that has challenged you.
So I have a question.
Okay, go ahead.
When an organism is dead, mfano kama a cow is dead, utapata during the daytime inatoa a lot of harufu, a lot of smell. Ndio, does it talk about temperature affecting the movement of molecules. Does it mean hiyo ina affect, as in hiyo the harufu, the smell yenye natoka kwa hiyo nini, is it ndio maana during the day it is smelling so highly than the evening ama the next day?
Yeah, that is true. You see, what happens is the smell of a dead organism is the same. Whether kuna baridi ama kuna joto, it is the same. But when it is very hot, particles will move faster. So this causes you to feel the smell, or even to smell that particular smell coming from that animal more, kwa sababu particles ina move haraka. So you will tend to feel like inanuka sana mchana kukiwa na jua kuliko usiku. But the real sense here is usiku kuna baridi. So therefore molecules zita move pole pole. Lakini mchana juu kuna joto, molecules zita move haraka because the kinetic energy of the molecules has been increased by the high temperatures. So si ati inanuka sana mchana, no, ni vile tu particles na molecules zina move haraka, so zinakufikia haraka, so unasikia ni kama inanuka sana mchana.
Okay.
Yeah. Okay. That's a good question. Another question, kindly. Well, if there is no question, then I think that will mark the end of the lesson. I'll see you tomorrow for the morning lesson, God willing. I want to wish you a very good night to you and your loved ones. Tuonane kesho majaliwa. Have a good night, y'all.
Night.